I'm a postdoctoral research associate at the University of Arizona. I did my PhD in France. Most of my work there was focused on the phosphorus cycling in the ocean. So I decided then to come to the Duhamel lab and pursue my phosphorus tree.
Hi, I'm Emily. I'm a first-year PhD student in the Duhamel lab. I fell in love with oceanography when I saw my first gray whale on the Oregon coast. Since then, I have decided to go from studying the largest creatures in the ocean to now exploring the smallest, well, some of the smallest, phytoplankton.
Today, we're going to dive into the ocean and study how marine invisible bugs,
by feasting on phosphorus, shape our visible world.
Before starting, take 2 breaths. Have you ever wondered from where the oxygen you're breathing is coming from? Trees and terrestrial vegetation provide half of the oxygen we breathe. But wait, from where the other half is coming from? To try to figure this out, let's take a look at the planet Earth. As we can see in this map, planet Earth is covered by continent, but a large part of its surface is occupied by oceans. That's why we call her the blue planet.
The ocean is not always that blue. Sometimes, and in some places, it's actually more green than blue. That green color is coming from a pigment called chlorophyll, which is synthesized by small organisms, invisible to the naked eye, phytoplankton. Phytoplankton in the ocean behave like terrestrial vegetation. When living their best life, they take up carbon dioxide from the atmosphere and release oxygen, thus providing the other percentage of oxygen that we breathe. So now, remember, one of the 2 breaths that you took at the beginning comes from the ocean.
Phytoplankton live around the world. Its chlorophyll pigment is used to look at its distribution. Here with this map, we show chlorophyll concentration over the global ocean. High concentration, shown in green and red shade colors, are observed in the northern and the southern ocean, but also along the coast. In open ocean areas, chlorophyll concentrations are low and are shown in blue shade colors.
To understand this distribution, let's take a look at what marine phytoplankton need to grow and happily proliferate. Similar to terrestrial plants, phytoplankton need nutrients, including phosphorus. In fact, all of life on Earth is built up on phosphorus. If we look at the phosphate concentrations over the global ocean, we can see that the concentration patterning is very similar to that of chlorophyll concentrations, where you have the higher concentrations in the northern and the southern regions, as well as along the coast, and you have lower concentrations in the open ocean. The open ocean areas, or in this map, the blue regions, are limited in phosphorus, so we call them P, or phosphorus, depleted. In our lab, we study phosphorus cycling and its importance to phytoplankton. In order to study phytoplankton, though, we either need to go to them or have them come to us. Living in a desert, we have decided to bring them to us. This is one of the incubators we have in the lab. We can change the settings to give our bugs the optimal conditions for temperature, light cycling, and light intensity, similar to their natural conditions. This incubator in particular runs on 12 hours with light and 12 hours without light to mimic the daily cycling. We maintain the culture in different size flasks that have a vent on the top to allow air to flow in and out. Here are 2 examples of phytoplankton called Synechococcus. This one is a species that grows on the coast, and this one lives in the open ocean. As you can see, the coastal Synechococcus produces a green pigment, while the open ocean Synechococcus produces an orangish pink pigment. To maintain our bugs over time, we need to ensure that no other bugs in the room, such as bacteria, go into the flasks. When we use the culture, like here where Kahina is monitoring the growth, we need to do everything in sterile conditions. We work under a laminar flow hood in which the air is filtered to make sure that the bacteria remain outside. Unlike the ocean, in the lab we can add all the nutrients needed for our phytoplankton to be as happy as possible.
Therefore, they live in a solution we call media, which has all of the nutrients and seawater a phytoplankton needs to grow happy and healthy. Over time, they use nutrients, and therefore the supply becomes limited. To make sure we keep them happily growing, we have a monthly phytoplankton buffet where we move the culture to new media with a fresh batch of nutrients. One of the critical elements for phytoplankton growth is phosphorus. To measure phosphorus in the ocean, we sample seawater. So here in the different test tubes, you can see different samples of seawater. After that, we add the phosphomolybdenum reagent, which is characterized by this yellow color. As you can see here, Emily is using a pipette to add that reagent into the samples. So the pipette allows us to add a small and a precise amount of the reagent into the sample. The characteristic of this molybdenum reagent is his capacity to bind with phosphorus and gives afterward a blue color. The intensity of that blue color would allow us to determine the concentration of phosphate in seawater. However, this reaction takes few minutes to occur. So in the meantime, let's watch Emily adding the reagent into the samples. Here we are. The reaction is complete now. As you can see here, we have the blue color appearing. Some of the samples are colored with an intense blue while the other ones are lighter. The intensity of the blue determines the phosphate concentration, so the darker the blue, the more the phosphate. Here, we're going to show you an instrument that allows us to measure phosphate by looking at absorbance values. Darker blue samples will absorb more light than the lighter samples, which will give higher values of absorbance. We use a clear plate where we can sample into each well. These plates contain 96 wells and will allow the analysis of 96 samples. This comes in handy when we have a lot of samples to analyze. Now that our clear plate is ready, we take it to the plate reader. After analysis, the plate is ejected and we can see our absorbance values on the computer. Phosphorus measurements can be carried out at sea on research vessels. To sample seawater, bottles are mounted on a circular frame and carried out into the ocean. We can trigger each bottle to close with the seawater inside from the desired depth. Once the bottles are back on the ship, the seawater is ready to be sampled and analyzed.
That wraps up the dive into our world as oceanographers.
Now, back at the surface, remember, the ocean is a breath of fresh air.